Hi, good morning, and welcome to the ZP Developer Zone. So we like to do this Developer Zone um, weekly just to really answer technical questions that have come in during the week or technical topics that we think we should discuss. Don't forget, there is the ZP Academy. It's a free, there's a couple of free courses on the ZP Academy. So if you're interested in electrochemistry, then that's a sort of a place to head over towards. And we have our webinars. This is an example of one of our weekly webinars. We also have them on Sense It All and some of our other technologies. We do have collaborations, especially in the UK and Norway, where the kind of funding allows us to participate. We do promote jobs, and we do have our ZP Developer Zone, and we do have workshops. So if people are really interested in sort of commercializing IVD, that's in vitro diagnostics, or they're interested in wearable biosensors, including CGM, that's continuous glucose monitoring, then we do have webinars, not webinars, workshops, and we have laid out some workshops, one a month for 2024, so those will be posted soon. I'll go a little bit faster and just say, in fact, we do have a workshop next week in Coventry. So this is all the workshops that we did in 2023. You can see there's a lot, so there'll be a similar time calendar for 2024. We do have some webinars next week. There'll be one on, and if you see a link jump up, by the way, I'll also put the link underneath the YouTube video as well, so you can kind of re-find these links, I would like to say. But that said, we do have a webinar next week called Sense It All. I definitely recommend that. And there's also something called Food Sense as well. So Food Sense and Sense It All are kind of cousins, can I say, because Sense It All is a platform that says, if you want to measure TAS, total antioxidative status, or nitrate, or pH, or you want to develop your own technologies, then Sense It All is the platform for you. And Food Sense is our application of Sense It All into the food sense industry. So those two webinars could actually be quite interesting. We are having a conference in 2024 in the UK on the commercialization of electrochemical biosensors. So I'll put a link to that website here as well. So again, I'll paste all the links underneath. It's probably worth backing up a bit and saying that actually, even though I'm going to talk about the measurement of analytes and carbon electrodes and gold electrodes today, there's a certain philosophy at ZP, and I think this is kind of makes us quite different, that in this world there are different types of, when I say end users, there's farmers, there's clinicians, there's patients, there's food technologists, there's people running industrial processes. There are people who want to measure something. And then there are people on the other side who, we would like to provide solutions to their problems, so we can be scientists, engineers, developers, entrepreneurs. And so the whole philosophy at ZP is actually to be able to get from the people who like solving problems to the people who have those problems as quickly as possible. I think what we've done at ZP is introduce the concepts of minimally viable products, which are something that is used quite a lot in the tech sector, and bringing that MVP, minimally viable product philosophy, across to some of this sort of deep tech industry. So if we're making biosensors or DNA sensors, which is a form of biosensor, then we're really into deep tech. But in deep tech, we don't talk about MVP, minimally viable products, quite so often. But at ZP, we do bring this kind of philosophy out. So today, when you hear me talking, even if I'm talking about carbon and gold electrodes, screen printed electrodes, I've always got this kind of commercialization hat on, because for me, there is, the real value in science is actually getting it out of the lab and into the hands of real people. And if you're going to do that, you have to it has to be scaled, and it has to be economic when scaled. So one of the things I'll talk about today is the cost of gold, and actually that's a real inhibitor at the moment for scaling, for example, assays based on gold electrodes, but I'll kick back to that. So the philosophy at ZP is to get from something in the lab to something that's actually in people's hands. So you'll see that our technology is based on this concept of hardware and reproducible screen printed electrodes and reproducible biosensors that you can start to put into the hands of users very early. And because we make everything web enabled, for example, if you've got one of our nitrate Gen 2 kits, if you're using it in the lab, every time you press upload, it'll go to a cloud called Julie, which is your account. And that will allow you then to understand, what does the signal look like? Is it looking correct? Do I see interferences? You can look at that raw signal and kind of make those decisions. And then it'll also allow you then to improve. So essentially what you can end up doing is you can end up passing technologies on to end users, having them to be your alpha and beta tester, but you're actually getting to see the raw data, so you can actually keep an eye on this. I think in the past you would have had to make something absolutely perfect before you could even give it to an alpha or beta tester. That's because you wouldn't have an insight into what was actually happening with that technology when it was out there in their hands. But the way we've closed the loop now, we can give technologies to end users much earlier on and see what's actually happening with their data. So I did mention a webinar on FoodSense, and I think in that webinar I will give a case study of actually how we're supporting. Even though I'm in the UK, a lot of the teams in Norway, we've got a lot of people using our food technologies in India, and we're able to support them because of this cloud this cloud capability. This is really a kind of a lean startup philosophy, which I don't think. I mean, lean startup to me has always been, I want to say, a thing. It's been quite a bit of my career in California, and so we were kind of used to this kind of MVP lean startup philosophy. But I realized that it's not something that is so understood necessarily in academic circles. Though one of the teams asking the questions today is in the US, so I think maybe there the philosophies are understood a bit more. So part of this philosophy about getting technology out of the lab and into the hands of the end users, I think is kind of reflected actually in this potential stack called Kate. So you can see that Kate is actually the same electronics and the same chassis as SensiAll, except Kate looks like, I would say, a sort of point of need device. Point of need, point of care, in vitro diagnostics. These really are just referring to hardware platforms where you're trying to decentralize test. So if we look at, COVID-19, you could send samples into the centralized lab and they would say, yes, you've got COVID-19. But then lateral flow strips came along and said, you've got COVID-19 because you could test yourself there and then. And that's an example of decentralization. So ZP, we're very interested in the decentralization of test. And that test, we can decentralize in the medical sector. We can also decentralize in the agricultural sector, which is probably a sector that really needs decentralization because there's no farmers are frequently running chemical, biological tests on the farm. All the samples have to go off and there's not much testing going on. And to a lesser degree, it's you could also argue the same in the food industry as well. So our potential stat actually looks like our SensiAll platform because in fact they're the same thing. It's just that the software is different. But that means then that you can actually translate from what was done in the lab to something that you can actually give to an end user so much easier. And the nice thing about the SensiAll platform is that we've already worked out the economics of this. So if somebody went to market with the SensiAll platform, we're able to give them 10,000 of these meters for $60 each, which is really nothing. I mean, this is a really, now, 10,000 meters. Well, to be honest with you, if a business is not able to sell 10,000 meters, it's kind of, it's not really a business. It's, it's a nice idea. So, in business, you have to be selling. If you're trying to make a low cost device, which many people who are trying to make these point of needs, often talk about, I want a low cost device. Well, you need to have high volumes to make low cost devices. So what's nice about this is the potentiostat translates beautifully onto SensiAll, and SensiAll then people can make business models about it because we've already got the cost out of the device. Here's a quick link. If the links jump up, then you can click those links underneath the video and find out about it. We are making all of our sensors compatible with SensiAll. I will discuss this a little bit more, but I'd also say come to the webinar as well. Something that's at the heart of what we do at ZP is the philosophy at the heart of us is actually this lean startup philosophy, how to get people to market or commercialization as quickly as possible. And then, the technology that we're basing ourselves on often is actually this kind of screen printed electrodes. Why? Because screen printed electrodes have demonstrated themselves to be very scalable, very low cost. But I do want to put a bit of a warning out about gold electrodes today, and that will be part of my talk. But I'll put some links to these screen printed electrodes. And then I have discussed that we have this SensiAll platform. You know, we have a spectrochemical version, electrochemical version, and a wearable version. But the one is most matured for, especially for academic colleagues, is the middle platform here. So the potentiostat that I mentioned earlier on is on the website, and I'm going to now sort of slightly change tack and actually go to the questions. That background is relevant to the question. So question number one really is about TAS sensors. This is total antioxidative status. It's also about pH sensors, also about nitrate, and they also asked about hypervalue carbon SPEs, which is a little bit out of family with the with those sensors, but we'll discuss it as well. And question number two is about gold electrodes, and I will discuss that as well. So the first thing is to discuss TAS sensors, which I will do. I looked at what they're kind of interested in, and I've made some comments. I'm going to make some comments on these items as well. So first of all, going to the TAS sensor. We've had to do a bit of a cleanup here. So we have TAS sensors, total antioxidative status sensors. You can buy packs of 100, and we have some hardware or the SensiAll platform to go with it. The nice thing about the SensiAll platform is it already has TAS integrated onto it, and I will discuss where that software resides. I suppose I was talking earlier on about this lean startup mentality, that in fact the application in order to run the TAS sensor on the SensiAll platform already exists. What the academic community needs to do in some ways is kind of spot what markets or what people could actually benefit from this kind of sensing, and then actually get these kind of sensors into the hands of these people, and then discover, you know, is it working with that particular person in that particular application? You know, because when I look at I think a lot of what happens in the tech industry is people are doing app development. Well, I think that kind of philosophy that, you know, what a lot of people in the academic community should be doing is probably app spotting and app development. So we have this TAS sensor, and I'll talk about actually that we've primarily used it. So you can, if you've got a Android phone, for example, scan that QR code. You can obviously pause the video, and then when you've got the software installed, you can actually scan this QR code, and it will change the app to be a TAS app, and I'll discuss that a bit more. Just a bit of a background on that TAS sensor, that we've put a lot of money and a lot of research into this TAS sensor. This video, by the way, is in one of the links that I've just put on a previous slide. Here I can see that we were using what's called the Gen 3 meter, but now we've gone to the Gen 4 meter. But it's a nice assay. You know, it runs and it uploads all the data to the cloud. In fact, she's got the results there. So TAS there is 1.07 millimolar, and I mean the way this kind of whole platform works, whether we're talking about TAS or nitrate, is, you know, you have a sensor, we have the meter, we have the app, we have the Julie cloud. Sensor goes into meter, sample goes on sensor, meter connects to smartphone via Bluetooth connection. App runs, app actually uploads the data to Julie, and you get a result on the phone. But in fact, what you also do then is get the raw signal in the cloud, which is great because you can actually then share it with yourselves. You don't need to download it and share the data that way. You can actually just share access. You can also share it with people like us, which means that if you're trying to troubleshoot something, it's much easier than to otherwise have to send files, etc. So it's got quite a workflow, let's say, associated with it. But for me, the previous video, we were actually doing TAS in plasma, total antioxidative status of plasma. I'm not sure if that's what these people are trying to do, but for each different matrix, there's going to be new problems. So there's plenty of science to be still done. On the pH sensor front, I kind of noticed the pH sensor you were organising. I were thinking of ordering. I would recommend the ZP hypervalue pH sensors. just a side note as well is that we're bringing a calibration-free pH sensor also to the Sensi All kit as well. I've got a link here to that as well. So I suppose one thing I should say about the TAS, the pH, and well, first of all, let's talk about the TAS. The TAS sensor, the total antioxidative status sensor, is a one-time use sensor. You put the sample on it, you get the results, you dispose of the sensor. It's the same for the nitrate sensor. You put the sample onto the sensor, you run the sensor, you get the result, you have to dispose of it. pH is a bit different because if you, for example, just get the pH sensors off our website, and I'll just sort of back up slightly on that, you can sort of you can reuse those pH sensors. Oh, I know why, because I'm using the wrong keyboard. So if you get these ones, you can reuse them. Now, how long they'll really last? This is always a question I get, because, you know, whenever something's wetted, there's two sides to that coin. There's the thing that's being wetted, and there's the solution that's wetting that. So with these solutions, I can never predict how long things will last, because I don't really know what people are going to do with the sensors. See, when you've got something like a temperature sensor, and a temperature sensor is actually behind like a polymer barrier, and you stick it into the sample, you get a temperature, you take it away, you wipe it down. Those kind of things. The sensor's not really in intimate contact with the sample. But with biosensors and electrochemical screen printed electrodes, they're in wet contact with the sample, so you can't really give a general comment on, well, how long will they last, because it really don't know what people are actually going to be doing with these things. But these pH sensors, we use them quite a bit. If we're going for the calibration-free system, then it's a one-time use as well. One of the things at ZP that lots of our sensors, because we do this kind of IVD work, this in vitro diagnostic work, it's very hard to make a low-cost, simple-to-use system where the sensor is then recycled, because you have to then prove that it's actually, that sensor is going to be cleaned and it hasn't changed during the cleaning process. So unfortunately, you've got to put the sample on there, you get the results, and then you have to dispose of the sensor. And I can see that, if we're really going to decentralize, let's say, hospital systems, it's the way it's going to go. Because you can't be asking patients to clean down sensors. It's going to be an absolute nightmare. There's too much manual work, and they'll put a lot of cost in the instruments. The nitrate sensor is the same. You put the sample on there, you get the results. You have to dispose of the sensor. We do have a kit on nitrate sensing. This is just a bit of fun again. This is the same app that you can download from the App Store. And if you scan this code from the app, then you'll find the TAS sensor and the nitrate sensor. So as a side note, a lot of our technology is designed for that sort of idea that there is an end user and there is you. And we're trying to develop things by having either potentiostats that look like products, customer-facing products, or screen-printed electrodes that have a high degree of reproducibility, so that actually you can get apps done a lot quicker, or applications for these kind of people much quicker. So there is a Sensi All webinar next week. Here's a link. If you sign up and you can't make it, don't worry about not being able to make it. We'll send you a copy of the webinar as long as you're able to attend. I suppose it's worth saying about this Sensi All platform is it's very elegant to look at, but it also has a very some really nice features like LED system that kind of show that the system is on, the system's made Bluetooth connection, the system is running the assay, and the system has uploaded the data. So it really sort of takes people through a nice experience. It's got some very nice hardware. There's a video running in the top left-hand corner that will show in a minute. This is one of our engineers running a system where they'll show that the data. Now, when you look at my hand here, I talk about the way that you can turn it on. The fact is, you don't have to slot. No more slots, because slotting, the problem with slots when you have to slot these screen-printed electrodes into devices is those connectors wear out really quickly. And slots are also a really good way of getting ingression of liquid into the device. That's something that kind of happens quite a lot. You can see here that we can just click it up. So there's a much nicer user experience. So whether you're using this hardware as a Kate potentiostat, or whether you're using it as a Sensi All platform, It's very elegant. You see the way that breaks apart like that. So if you were doing soil measurements and the farmer got soil on there for some reason, or it was a nurse and there was for whatever reason a bit of bodily fluid on that, you can just take it apart and clean it. In the other video that's running at the moment, the engineer is just talking through the app, but she'll do an assay in a minute. And when the assay is completed, she's going to put the sample on. When the assay completes, she'll actually talk about the data has actually already gone up to Julie, so she can see the raw data in the cloud, which is really a benefit, both from your research perspective, but also a real benefit when putting real systems in the field with real users. And then the last little video here is just this SensiAll app, which you can download if you scan that QR code, which is in one of my slides earlier on. And I'm just going between these different QR codes, just showing you that you can change the assay really quickly. But the nice thing is if you're in academia, you're not excluded from the raw signal. You get the raw signal. The user on their phone will get a result, but you will also get the raw data as well. So you can sort of, troubleshoot things and find out what the hell's going on. So here's some of those QR codes: chili, ginger, nitrate, TAS. You can scan those and see how that runs. Julie as a database, Julie is no, let's say, flash in the pan. This has been almost 10 years now in development, more than 9 years. So we're going to get up to a 10th year. Julie is a cloud database system, cloud data management system, specifically tailored for electrochemical data. So it's the only one in the world that's commercially available. It stores data. You can visualize data up in it. You can do data processing, data acquisition through that SensiAll platform or through the Cape PotentialStat. It also has an API built into it, an application program interface. So API goes back to our whole conversation really about the lean startup mentality. APIs is really the glue that makes our modern world work. Banking systems talk to each other through APIs. Contactless pay works through API. Booking, airline tickets, everything works through API. And so we have an API built into Julie. So it means that people can actually then make a call upon Julie and get data into their clouds, because then they can sort of, in the end, API does allow people to monetize technology. And if you talk about sustainability, really things become sustainable when there's actually enough money being made by that company that they can actually then go on to pay the salaries of people, and then people can sustain the solution that's therefore being provided. So Julie, all the data ends up in Julie. Julie's great because you can share the data with internally with your team, or you can share it externally with people like ZP if you want troubleshooting support. I'm going to change tactics now and come off. I've been on a lot of deep tech, sensors, hardware, cloud capability, up to the cloud. I do want to talk very quickly about gold electrodes now as well. So let me go a little bit faster. Well, not faster, more efficiently. So somebody's interested in our gold electrodes. They want to make a DNA test from them. This is, so you have a gold electrode, you use a self-assembled monolayer, you put a single strand of DNA on there, which I would sort of describe as the capture probe. Then you have another strand of DNA, which is the target probe, and these two things bind. And you can sort of show that binding is happening by using a mediator like methylene blue. Because when you've got a single strand and then it forms a double strand, you end up forming obviously a double strand. And in those grooves, methylene blue can, I want to say, intercalate. And that means then that the methylene blue signal increases, or changes at least, and you can look at it using square wave voltammetry and get a signal for it. So you can see that binding event. So that sort of science is, something that's, there's a lot of literature precedents for that. The problem with gold electrodes, especially the ones that the person's interested in, is that ZP started. We're in our sort of 10th year now, so these were literally the first products we ever made, and they were fairly standard at the time. But over the years, we have learned something about them. They're too big. They're physically too big. What I mean by that is, if you were watching the slides much earlier on, I was talking about that lean startup. If something's too big, we can't make it economically at scale, and therefore the costs You can't commercialize off that platform because, in fact, the platform is inherently too expensive. And part of the expense comes from being too big. I'd also say this is that there's also a squeeze on this material because we're making it from ceramic. A few of the sensors that are going to the R&D community are made on ceramic, but ceramic prices are actually getting quite high. So they're too big, the materials are too costly. I put a bit of a flippant comment here, they're too gold. Gold is really spiraling in price, and I really don't think over the next couple of years that gold prices are going to come down. In fact, when I think about it a little bit, when you've got economic uncertainty in the world, gold prices tend to go higher because people tend to sort of buy gold. Gold is a sort of fairly scarce resource. There's a lot of demand for gold from the electronics industry, you know, from the jewelry industry or the luxury industry as well. And then there's people like us who want to make screen printed electrodes. You know, kind of, you know, gold is not a material that's readily available, and there's a lot of people trying to fight for it. And I'd also describe gold as a little bit unsustainable, to be honest with you, both because of the competition for the material, but also, you know, when you look at where it's mined, it's not coming from very sustainable resources, I would say. So, but the links to those electrodes are here. Now, at ZP, we do have a bunch of screen printed electrodes that I can recommend, and I would recommend this Hypervalue Gold electrode. So it's not this link, it is this link. Now I'll make some arguments about why. We do use that Hypervalue electrode a lot in our own research. I think it's something that, I haven't said it in this particular webinar, but the biggest users of ZP screen printed electrodes is ZP. We do it for our own products, and we do it for our own for our clients as well. And then they make their way onto the web store, and academics and startups and entrepreneurs buy them and use them in their research and development. So we do actually use that Hypervalue electrode, gold electrode, quite a lot in our own research, and testing here at 5 nanograms per mil protein, 25 nanograms per mil protein. We're obviously using EIS here, impedance spectroscopy, electrochemical impedance spectroscopy, but I would say EIS, square wave voltammetry, differential pulse voltammetry. If you look at an assay with those three ways, they all essentially work, and so you can sort of translate this into being square wave voltammetry as well. What's nice about this is the reproducibility of the electrodes here, because there's actually 3 electrodes here being tested at the same concentration, but because they're so reproducible, you can't realize that there's actually 3 electrodes under there. With this one at 75, you can see that there are 3 electrodes under here. So my problem at the moment with gold is really the pricing. I looked at the pricing yesterday, and it's sort of $2,000 per ounce. I'm a bit of a metrics guy, so I put that into it's $71 per gram, just to help me understand the numbers a bit better. And I did, so it's expensive, and it is. I mean, since ZP's been around, it's essentially gone from at least 1,300 euros. Today we're at 2,000 euros, so it's really kind of spiked up, and I don't see it coming down anytime soon. There's also a bit of an issue. I mean, I think many people are being asked these days, at least in Europe, to have sustainability. As if they're going to try to get grant funding, they have to kind of demonstrate sustainability. I'm really not sure that sustainability, by the way, I only use these images for educational purposes. I just want to make that disclaimer. But when you look at this, you can sort of see where some of this gold is coming from, and I'm not really sure that it's necessarily sustainable, and I suspect that a lot of these gold mines are causing local pollution incidents. I don't want to name names here, but they do use cyanide as part of kind of gold extraction, for example, and I know it ends up in rivers and causes pollution to the local communities. And if just Google gold mines, and you'll see that these are kind of large scars upon the planet. So in terms of recommended gold electrodes, I do have a recommended gold electrode. Overall, I'd like to make the argument, and I'll make it in a bit, that actually, if you've got the choice now, I would choose to go for carbon. Look for carbon. Look for chemistry that will allow you to couple to carbon, because it's so much lower cost. But if you are going for gold, then I will make this recommendation, this gold electrode. If you've got Potentiostats these days, I don't know what potentiostat you have, but they generally have banana plugs on them, and they're either in 2mm or 4mm. Banana plugs measure the diameter of them. A lot of people in the US at the moment are actually using potentiostats with 2mm banana plugs, so there's also a connector for it. One of the biggest things I'll say about gold electrodes is, if you're getting poor performance on gold electrodes, I mean, I'm sure people are doing this, but we always have a cleaning step before we do the functionalization step. Before you make that self-assembled monolayer, you do have to kind of clean it. We clean it, we verify that it's clean, and then we do the, when I say verify it, we do that by cyclophotometry, and then we do the attachment of the SAM layer there. So as a side note, gold versus carbon, I mean, we do understand there's chemistries for both. You know, we do use gold electrodes. In fact, I was talking to one of the engineers yesterday who really likes our gold electrodes, because I suppose, you know, they've matured so much. But then with my economic hat on, and also my kind of sustainability hat on, I would actually wish that people would go for carbon electrodes a little bit more. But this kind of self-assembled monolayer works perfectly well on our gold electrodes. But I'm just suggesting the hypervalue gold electrode. And the reason I'm suggesting it is because we use it so much. And the other ones, you'll see that our pricing is extraordinarily high, and it's really reflecting the cost of gold at the moment. Something that you'll notice, I did recommend a little while back in this webinar that people sign up to come to our Sensio webinar, and maybe even to come to the Food Sense webinar. What you'll notice in our webinars is that we're not doing any calibration when we're doing our assays. It's both because, in the end, I mean, I have a lot of experience in assay development, and, you know, ZP is an ISO 13485 business, so we're very used to making sort of point of care, point of need medical diagnostics, and you can't have calibration on products that are necessarily going to final end users. So you'll notice that when we do those webinars that we don't do any calibration. And the reason being is, this applies to our carbon electrodes and our gold electrodes, but ZP does, I would say, more characterization or more OQC, outgoing quality control, to make sure that these electrodes that we're shipping to you are actually reproducible. If you can make reproducible screen printed electrodes, then you don't have to do in-field manufacturing to the, in-field calibration. Often, when you see scientific papers presented, people will normalize data. They'll take a sort of signal on the electrode before adding the analyte, and they'll take a signal after adding the analyte. The reason they're doing that, then, that normalization, is because they know that they've got reproducibility issues on the screen printed electrode, and they're trying to normalize that out of their data. And then when you sometimes see products on the market, when there's a sort of, or products that are talked about in scientific papers and they have a calibration step, it's because actually the fundamental electrodes are irreproducible. You really want your manufacturing reproducibility to be sort of less than 10% to have a useful point of care device, and that's why we look at our relative standard deviations and make sure that these sheets have less than 10% standard deviation between them. And so when you look at, when we take one of our screen printed electrodes, this is not work done on the same screen printed electrodes. We're using something like it might be about 18 screen printed electrodes in order to get this data set. So we test low on 3 electrodes. We test also quite low on 3 electrodes. We're testing low on 3 electrodes, testing medium on 3 electrodes, testing higher on 3 electrodes, and testing higher on 3 electrodes. Here we're using voltammetry. But it's just to kind of talk about the reproducibility of our screen printed electrodes. And just to sort of say that, look, you know, I understand people want to go for gold. We use it ourselves, but carbon, I've put a paper here that we've, and I'll put a link to it. This is a way of functionalizing carbon electrodes, and I would recommend that people start seriously looking at carbon electrodes, and maybe, even if you've had a long history of using self-assembled monolayers, gold ain't coming down in price. Gold is not, I would not describe gold as necessarily a sustainable material. Whereas I think the accessibility of carbon and the demand for carbon is very different. So I would describe that as actually more sustainable. And the coupling chemistry to make immunosensors, for example, on carbon is not that hard. It's quite, I'd almost describe it like click chemistry. This is just a comment I made earlier on. I realized when I was talking about cleaning gold electrodes, what we do is we tend to clean the gold electrode, then we check it by cyclophotometry to make sure that that electrode really is clean. Then we start assembling the functionality on top of it, and we still check by cyclophotometry that we are functionalizing that surface, and we're testing it with. This is some work that's actually in that paper where we actually used, it was a carbon electrode, but we did use differential pulse photometry in that work, and we got some really nice signals, and I do recommend that chemistry quite a bit. So if I was to sort of summarize today, I think I'm sorry that the pricing is so high and the pack size is so high on those gold electrodes. There's an economic fundamental reason behind this. And the only one we're really doing small pack sizes on the hypervalue carbon electrodes. But I would actually generally recommend that you actually went for the carbon electrodes. We're doing sheets of carbon electrodes for like €250 for 300, and they're high quality. It's just simply because we're not using gold. Gold is expensive. So I would recommend that. So my advice is go over to carbon. I mean, when I looked at the kind of carbon prices, I know I said gold earlier on was like about $2,000 per ounce. Carbon is something like $1,500 per ton. Carbon's like 1/28,000th of the price of gold. So I would wish that people would go into gold. If you don't have the kit to do EIS, we do have a EIS potentiostat. We do have hypervalue carbon electrodes. We do have the cables that you need, and we do have the connectors as well. So in summary, the person was very interested in our TAS pH, our nitrate sensor, and our hypervalue carbon electrodes. Just to say that those TAS sensors, they are a single use. You put the sample on, you get the assay, results go up to the clouds, you can analyze those results, but they're single use. The pH sensors, I've put a few pH sensors up there, but the ones that you're looking at, there's hypervalue pH sensors that I'm recommending. You can reuse them. But if you get there's something that I've also linked to today, which is if you get the calibration-free pH sensors, those are single use. The nitrate sensor is also single use as well. And then I've talked a lot about the hypervalue carbon electrodes. You were trying to get a pack size of 10, which is, I think, just too small. I could just get a pack size of 300. It's just sort of better economics, to be frank. And then on the gold electrodes I'm recommending, the hypervalue gold electrodes, it's a personal apology that we can't do. The pricing is very high on the other ones, and the pack sizes are very high as well. There's simple economics here that we just can't. You can't waste any gold, so you can't make small batches. We have to, but with the hypervalue gold electrodes, because we sell so many, we're in constant production on those, and those are the ones I recommend to you. And I suppose with that I say thank you, and I sort of wrap up because I realise I might have spoken for. I'm not even sure. Close to 35 minutes by the looks of it. So if you've got any questions, don't hesitate to reach out to us at ZP. You can see we put a lot of effort into answering questions, and I wish everyone either a good rest of the day or a good day. Okay, thanks very much.